So good afternoon. My name is Rune, and I'm from the Bioing group at the Technical University of Denmark, and we specialize in applied enzymology. And specifically, this project has been about the production of different human milk oligosaccharides. The outline of this presentation, I'll talk to you a little bit about human milk oligosaccharides, structures and properties. We'll explain how we make them, the sialidated HMOs at the Bioing. I'll tell you about this little study of in silico trans-sialidases discovery, and I'll talk about the verification and take you through the conclusions and perspectives. So, human milk oligosaccharides. This diagram is from an excellent review by Bode, where he talks about all the different properties of human milk oligosaccharides. There are these common building blocks, which we've seen again and again these past couple of days. And it's backbone structures, which are then sialylated or fucosylated at different terminal positions. Why do we want to make them? Well, to have happy babies, basically. There are all sorts of postulated beneficial effects. Prebiotic effects have been known for a long while, but there are several other effects, adhesive effects to microorganisms, can in that way work a bit like antibodies, so the pathogens recognize these organisms, think it's a cell wall, and gets flushed out. Brain development, sialic acid is a building block in the brain, and there's some strong evidence for sialic acid contributing to brain development. So the setup that we use, we have is now fairly well documented in literature as well. We use the side stream product from the dairy industry, CGMP, casein glucomacropeptide, which is sialylated, and then we transfer these sialic acid groups from the CGMP to an acceptor molecule, in this case lactose, and yeah, here we produce 3' sialylactose, which is a genuine HMO, but recognize it from the previous slide. There is the risk that our product will again be hydrolyzed to free sialic acid, lactose, and that's obviously a reaction we want to make sure doesn't happen. Also, the substrate can be directly hydrolyzed, obviously. And yeah, as said, we do applied enzymology, so of course we're using enzymes to carry out this reaction. And there's one known native trans-sialidase, and that is the trans-sialidase of Trypanosomus cruzi, which we were briefly introduced to earlier. This is the pathogen, and this is its host. The pathogen uses the trans-sialidase to cover itself with human sialic acid residues, in that case hide from and protect itself from the human immune system. So, there's the problem that this sialidase is from a pathogen, so when you want to apply it to make human milk oligosaccharides as additives for infant formula, you don't really want to sell it as something that has got anything to do with the pathogen. Another point I want to make is that it's not a transferase. It uses a non-activated substrate, right? So it's from this sugar chain. It's not a CMP activated substrate. And yes, it is a virulence factor, as we just covered. So we go to its close cousin, the sialidase of Trypanosomus rankeli instead. This is a non-pathogenic organism, and as studied by Paris and colleagues, demonstrates how by selecting a couple of sites for mutation, you can actually infer some trans activity to this enzyme. We in our group have put a big effort into optimizing this enzyme through several rounds of directed evolution and created this TR13 enzyme, which is our workhorse normally in our setup. So, and just to say that this is not an easy thing to do, my colleague who also did the TR13 directed evolution study has tried to convert trans activity to micromonospore viridifaciens sialidase, and he observed no trans activity whatsoever in more than 200 constructed mutants, which were selected from a pool of, I think, 1,800 different mutants, site-saturated, mutagenesis in many positions. So that's not straightforward to infer this trans activity. Well, we're left with the chromosomes Krutzi, knowing that this TR13 is also. We'd like to know more about trans-sialidases, because this trans-sialidase of Wrangeli that we made the TR13, all these mutations are basically also found in the chromosomes Krutzi. So yeah, we are making a new trans-sialidase which originates from a non-pathogen, but we're doing it by reversing it towards the pathogenic enzyme, which is also, that could at least be a threat to the application of the enzyme. We have trouble since we don't have any. We want to find new trans-sialidases, which is difficult because there are no good screening assays. Most of the enzyme discovery assays we use depend on a breakdown product that is some sort of reporter product as well, and we can't get that with the trans reaction, since we're not interested in breaking down anything, only the transfer. It's hard to find any conserved amino acids in a database of trans-sialidases, since we only have the one. So what do? Well, first of all, we reacquaint with the trans-sialidase of chromosomes Krutzi and find out what can we actually tell about this enzyme. It has got a conserved active site, which it shares with all the sialidases. So it is a sialidase which has then developed into a trans-sialidase. And one of the things that is immediately visible when you look a bit at this structure is that it's got this hydrophobic pocket or aromatic sandwich constituted of a tryptophan and a tyrosine above the active site. So that made me hypothesize that the family of sialidases is extremely conserved. It can be speculated that a trans-sialidase could have developed in parallel with the TCTS, inferring an aromatic sandwich in a similar position as it is found in the TCTS. This struggles a bit to be an actual hypothesis because it's pretty difficult to disprove. However, if hypothesis 1 is correct, sialidases with an aromatic sandwich above the active site will be a good place to look for candidates with trans activity. So that is what we tried to do. I established this database of, or just downloaded from GenBank, 2,900 genes that are labeled as sialidase, neuraminidase, such like. And then I constructed a search motive to try to find these sandwiches. And to some extent, we're lucky that this tryptophan is positioned only 2 amino acids away from this highly conserved arginine of the sialidase active site. So we exploit this luck and create the search pattern here where we have a tryptophan, tyrosine, and phenylalanine, so an aromatic amino acid 2 positions away from this arginine. And yeah, this RDR is not completely conserved, but also relatively conserved among the sialidases. So this is our search pattern. If we look a little bit closer on the active site and investigate these different amino acids which are all involved in the activity of the enzyme, we see that they do all have a function. So we decided to include them all in the study further on for selection of our candidates. And this is what we did. There were 15 candidates that shared this motive, and all of these we then continued to make 3D homology modeling of using HH-PRET or CPH models, and then I simply aligned the active site residues with the comparable residues from the TCTS. And this was partly to make sure to see whether it has an opposite aromatic residue, and if any of these, or if all of these active site amino acids model well. We can see that 4 of the candidates did model well, so all of the active site residues are in fairly close proximity to the TCTS enzyme. And if we pay a little bit close attention to the CLH, we can see that it has actually got an aromatic amino acid opposite the tryptophan, constituting an aromatic sandwich. This is where the organisms are from, so 4 organisms and relatively large enzymes, but in the same range as the TCTS. We'll use the abbreviations from now on. Again, here's an overview over the 3D alignment. We can see that some of them do not align perfectly, but a correct amino acid would be just shifted 1 position. So this could be down to a modeling error that is fairly common with these 3D homology models. We can see that these 3 candidates here do not have an aromatic amino acid opposite the tryptophan. That, however, has the CLH, and you can see the aromatic sandwich here. This is a substrate, sialilactose, in the active site. So the 3 remaining candidates are also included because they all have a very closed active site. This model here shows the viridiphasians, that's the one in color, compared to the T. cruzi sialidase, trans-sialidase, and it's very obvious that it's got a very, very closed active site structure. We include them to see if this is enough to infer the trans activity that we're looking for. So, decided to express these enzymes in E. coli using a T7 promoter expression vector system, auto expression 30 degrees overnight, and we purified the enzymes to make sure that we could actually allocate our activity to our enzymes. And then, of course, decided to apply it to the Bioeng Ciali HMO production setup here. And it's worth noticing that we just use 30 degrees and pH 6.4. This is our standard setup with 100 grams of lactose and 40 grams of cGMP, basically keeping a high level of lactose to drive the reaction towards our desired product. So, did that, analyzed the samples by HPLC. Oh, that's something wrong with my presentation. Oh, sorry. Okay, we'll work around it. So this is typical what we see with the sialidase. This is where the products are supposed to come out. And we can see that there's basically only free sialic acid, a huge peak from the lactose, obviously. To be fair to this enzyme, there is actually a little bit of 6' sialic acid, which is, so there is a little bit of trans activity. However, this is the CLH, and we can see here that there is a huge peak where the trans product comes out 3' sialilactose. So we have trans activity. And also, we have a second mystery peak, which we, of course, went straight ahead to analyze, to figure out what this is. And the first thing we did was run mass spec on it to establish that it is some sort of sialylated lactose compound. And then we treated it with beta-galactosidase and figured out that, therefore, the sialic acid group must be on the glycose part of the lactose, since the beta-galactosidase would not recognize the substrate otherwise. This was also confirmed by NMR later. So, if we look at the CLH, of course, back to the original study, the plan, and we see that we actually built up a nice level of product here, and it's actually relatively stable. We also ran some overnight reactions, and in the end, the enzyme will break down our product as well. But we reach a nice level, and we would be able to stop the reaction at any time and recover some product. We also see this new compound, 3-sialilactose, also building up to a relatively nice level. It's also worth mentioning that this reaction is not optimized. This is obviously done with the standard setup. The other thing would be next step. Yes, and we compare that to the other candidates, and so they don't make very nice diagrams. Basically, they are all very much more hydrolytic. There's a little bit of product formation, but it's better shown in this this table. But again, it is very, very obvious that CLH has got way better trans activity than the other candidates. So, gift you the trans-sialidase of Haemophilus parsuis, which we're very happy about having found. And if we draw some other conclusions, we can say that yes, in this database of 2,909 sialidases, we did manage to find a trans-sialidase. It was identified by rational sequence analysis strategy, and it's the first confirmed bacterial trans-sialidase. There are, now said that Chicrutis is the only trans-sialidase. It has got a couple of pathogenic cousins as well, Turbosomus congolese and Brunei, that are also, they're just very, very similar. This enzyme has a 30% sequence identity to the other trans-sialidase. We have a new product which we have identified, and all 4 candidates showed some trans activity. If we hold that up against the couple of hypotheses we put up, we can say that yes, it does seem that this aromatic sandwich was a good idea to look after this aromatic sandwich. But of course, I can't say that there are not a trans-sialidase which have a completely different mechanism. Yes. Perspectives. It would be really nice to do some crystallography of this new enzyme to see that it actually is the proposed aromatic sandwich in the position where we expect it to be based on the model that confers this activity or helps it. Hopefully we could draw see some common traits and use that to further improve our TR13 or identify more native trans-sialidases. And why not find some other trans-glycosidases? We just need to find some more sandwiches. So that, I would like to thank my colleagues, my supervisors, and all the other people here on the HMO project, especially Carsten, who forced me to follow this little idea instead of a couple of other pressing issues. So with that, questions, please. Thank you.
Thank you very much. Yes, question.
So I have a question. The new compound you found, the sialic acid attached to glucose, does that actually occur naturally as an HMO?
No, that was the thing. It's not an HMO. It's not, at least not been identified as an HMO. But there's some other interesting. There are many interesting details, but now I focused on the good story. Parsuis is a pathogen as well, which make this not very applicable straight away, but it's a pathogen in pigs. So it could be, if we look into the glycosylation pattern of pigs, that this compound might occur there. And I have seen data, not HMOs but PMOs, I guess, that they also make sugars in their milk. Elephants does it as well, and we're looking into some different other strains. What are they called? Mammals.
So have you used other receptor substrates rather than lactose and cell bias, perhaps?
Not for for the TR13 we've used a whole range of glycans as acceptor molecules. Also some of the HMO backbone molecules. We are also working on, and we have managed to, from smaller building blocks, create some of these backbones as well. But that is basically the whole concept of the overall study.
So is there a reason for going after sialated ones as opposed to the fucosylated ones?
What, sorry?
So is there a reason for going after the sialated ones as opposed to the fucosylated ones?
We are also going after the fucosylated ones, so we're basically attacking on all fronts. And this is my little my niche. Well, I've done the gluconate backbone projects as well. touched the fucosylations yet. And also, we're lucky that this trans-sialidase exists. That's obviously one of the clues for going in this direction at all. I mean, glycosidases could be very powerful because they don't need to use these activated substrates. So in all sorts of biorefinery or biomass exploitation, these enzymes would be nice to find.
Okay, very nice. Any more questions? Yes, please.
I had a question about the mechanism. So, do you know anything about the mechanism? Is there a covalent intermediate? And
Yes,
also
yes. There's that's all. Yeah, we can maybe see it. I haven't got that model, but yes, it is covalently bind. The sialic acid residue binds deepest in the pocket, make a covalent intermediate. The whatever substrate floats away, and then basically this intermediate is attacked. And we don't really know whether the sandwich is essential, and why that makes a difference. We think that it could be, preventing water from attacking
Exactly, that's what I was thinking.
the intermediate, but exactly how, we're not sure about.
Okay, very interesting.
Okay.
Otherwise, I have a poster if there are more questions, so please come and ask more. Thank you.